I anyways just start to introduce what microtubules, oh, okay, no, it's not. Microtubules are. So microtubule are cytoskeleton involved in processes like mitosis, cell motility, intracellular transport, and so on, almost everything. And there are dynamic biopolymers that undergo polymerization, depolymerization cycles, which are more prominent as at their plus end than at their minus end. Microtubules are built out of alpha and beta tubulin dimers. So you can see them here in green and yellow respectively. And they assemble into those tubules. The sides of those tubules you call protofilament. And those tubulin dimers have quite dynamic tubulin tails that are recorded to be important for the microtubule functions, but they are invisible in all high resolution structures. So cryo-EM and crystallography having hard times with them. They are also subject to a variety of post-translational modifications, so that Kirsten Janke coined the term tubulin code in resemblance of the nucleosome code. Yeah, they are also implicated in diseases. For example, a missing glutamylation is related to male infertility. So quite interesting. For microtubules to function, microtubule-associated proteins, short MAPs, are needed. They have a variety of functions. For example, they can help to stabilize microtubules. They can be motor proteins walking along the microtubules. They can bind them to other cell structures and so on. And as well as the microtubules, they are also associated with diseases. For example, neurodegenerative diseases. Their microtubule binding regions come in all shapes and forms. So here in the middle, you see the maps with which I'm working. And they're so basically they're microtubule binding domains. And what is also quite common for maps is that they have larger intrinsically disordered regions. Okay, here I'm going to show you some experiments using solution and solid state NMR. And so the solution state NMR, what I always will indicate with this, image is for labeled maps and unlabeled microtubule tails. And the solid state NMR I will use for the bigger system. So I will label the maps and use unlabeled microtubules or the other way around. And some of my solid state NMR experiments were done on this, at that point, prettiest magnet in the whole world. So first of all, how do I get even the microtubules? So we can purify labeled or unlabeled microtubules from HeLa S3 cells in cell suspension. And we are purifying them by polymerization, depolymerization cycles. So microtubules are depolymerizing at 4 degrees. So when you take the supernatant, you get the tubulins, and they're polymerizing at 30 degrees. So when you take the pellet, you get full microtubules, and then you repeat this to get rid of MAPs and other stuff of the cell and so on. So we obtain quite clean tubulin, and we also checked via microscopy if they're intact after magic angle spinning. So, and that seems to be the case. Okay, after we get microtubules, we can now study the interaction with MAPs. And first of all, I'm going to talk about microtubule-associated protein 7, short MAP7. That is a protein that activates kinesin 1. So you see it a bit in the figure. It is catching kinesin 1 out of the cytoplasm and helps it to bind. And it seems to inhibit other kinesins. And otherwise, it's a microtubule stabilizing protein that binds along the protofilaments and also seems to bundle microtubules. The microtubule binding domain is only a small part of the whole protein. So this year, a cryo-EM structure was published of MAP7 bound to microtubules. but this QIUM structure is actually showing an alpha helix shorter than the region that is recorded to bind the microtubules. And yeah, also what I did with solution state already. So I signed MAP7 via solution state just with, yeah, an array of 3Ds, shows that the alpha helix that MAP7 has might be longer than yeah, what they actually recorded in their QIUM data. They also claim in their paper that most likely MAP7 is binding longer, but with QIUM you have the problem of pseudosymmetry in microtubules that, yeah, makes it difficult to get electron density longer than a dimer. So therefore, we assume that there might be more binding than what was recorded. So to study this, we turn to solid state NMR, and we label our MAP7 microtubule binding domain and record spectra, yeah, for the flexible and the rigid paths, so scalar and dipolar, CH, and also NH spectra. Here I show the CH spectra. And we compare them to our solution state assignment and check which residues are unambiguously either flexible or rigid in our NMR. And yeah, by plotting them, we get the following correlation. And comparing this to our solution state NMR helix, we see that the N and C terminus still, so in blue are the residues that still seem flexible, and in red the ones that still seem rigid. And we observe that the N and C terminus still are quite flexible, while the region in between that is alpha helical in solution also is yeah, more rigid and solid, and even residues continuing after this alpha helix have more rigidity. What might mean that, yeah, our MAP7 is binding with this whole helix. And yeah, this is therefore kind of our model right now, but this is just the crude PyMoles, yeah, idea. Furthermore, we studied the tail interaction with MAP7. So we made a peptide of the tail, of the C-terminal tail that is, as I said in the beginning, dynamic, and added it with a titration to our map. And we observed, so this is solution state data, we observed shifts in two regions around residue 85 and residue 140. And also by doing solid state of labeled microtubules and adding unlabeled MAP7, we see decrease in signal in both alpha and beta tubulin. So the assignments of this were done by Yansang Lu, the PhD student who started the microtubule project in Mark's group. And yeah, so therefore we assume that there's a residue-specific interaction. And yeah, this is especially in two regions. By comparing the alpha and beta C-terminal tail, so beta in yellow and alpha in green, we see that the interaction seems to be similar for the first probably binding region, but less for the second, especially for the beta C-terminal tail. And yeah, so this is a weaker binding, what we also yeah, verified with techniques like fluorescence anisotropy or ITC that I don't show here. Yeah, to conclude for MAP7, MAP7 binds with an elongated alpha helix to microtubules, and there are two regions identified that seem to interact with both the tails, while the second one shows a stronger interaction with the alpha tail. So following up on this, I want to talk about the second protein that we studied. So it's CCPN that is totally different again, tau. So tau stabilizes microtubules, and it forms, yeah, it is well known for its correlation with Alzheimer's, where it forms neurofibrillary tangles. And yeah, there it is not binding to microtubules anymore, what destabilizes most likely also the microtubules. It is an intrinsically disordered protein, and yeah, we are using the tau K32 construct that compromises the 4 repeat regions, R1 to R4, that are the microtubule binding domain, and also the flanking P2 and R prime region. So far, one of the best structures is this QuAEM structure from 2018 that is just one of the repeat regions. So as for MAP7, we also labeled tau and yeah, did some solid state with it by incubating it with the microtubules. And we recorded scalar and dipolar spectra and checked how this is in correlation with our solution state assignment. Yeah, that gave us the following pattern where we basically see that the flanking regions P2 and R prime are more flexible, while R prime already shows some rigidity. And yeah, the repeat regions show more rigidity, while the R1 is still more flexible than the other ones. So here's a meta-interference model from Boutsakis et al., where I plotted those correlations on it. And yeah, that the R1 is actually more flexible than the other repeat regions is in line with the suggested simpler mechanism of binding of tau to the microtubules. Yeah, as for MAP7, we furthermore also studied the tail interaction, and we titrated our C-terminal tail peptides to tau and observed what is happening. And yeah, basically we see in all 4 repeat regions specific shifts around histidines prior to the so-called PGGG motif. And yeah, so we assume that the tails are binding to that. And by doing solid state NMR, so looking at the other side, looking what the microtubules are doing, we see decrease in signal intensity, especially in the alpha tubulin tail. So yes, especially in the EGE motif of this tail. For the beta tubulin tail, there seem to be some decrease, but it is not as prominent. So therefore, we assume there might be more interaction with the alpha tail. That is also in line with what we see in our solution state titration, where we get stronger shifts for the alpha C-terminal tail. The binding affinity for this interaction seems also to be in the higher micromolar range. Yeah, so our model is now that tau tail binding is in the regions of the EGE of the alpha C-terminal tail, and with those histidines of the, yeah, this histidine lysine is normally of our tau. So to conclude, for tau, tau binds to microtubule with all 4 repeats, but not with the surrounding region, while the R1 seems to bind less tightly. And the region prior to the PGGG motif seems to be involved in the tail binding, while the interaction with the alpha tail is more significant, and especially at the EGE repeat. Yeah, and finally, I have a third microtubule-associated protein that I want to quickly introduce to you. So CAMSAP. CAMSAP is a protein that regulates the microtubule minus end dynamic. So the minus end is the less dynamic end of the microtubules that is often stabilized or anchored. And its end specificity is mediated by the so-called CKK domain. That's the microtubule binding region. Yeah, you can see it here. It is quite globular and has a high beta sheet content, what already differs it largely from the other 2 maps that I am studying. So map 7 with the high alpha helical content, and tau that is intrinsically disordered. And also the binding mode of CAMSAP is really different from the other 2 because it's not binding along the protofilament, but it is binding in between 4 tubulins. Yeah, so here my pre-PhD student already started to look into the interaction by labeling the microtubule and adding the CKK protein. And he actually observed shifts in the solid state spectra that clearly show that there's some interaction. Also from the QuiUM data, it seems feasible that the tails are interacting with the CKK. The thing is that CKK has a long C-terminal tail that is also flexible and therefore not visible. And therefore, this information is, of course, not obtainable by QuiUM. So yeah, as for the other proteins, I also did a titration with the C-terminal tail to the protein. And I basically observed some shifts, especially at the C-terminus, but also an increase in intensity in the later residues due to the binding. The binding affinity of this is a bit stronger than of the other two maps. And yeah, by plotting the intensity, so this is just with different salt concentrations because it's an electrostatic interaction, so good checkup of the interaction. So, but we basically see an increase in intensity in those residues at the C-terminal tail. Yeah. And therefore, it seems quite likely that this tail is interacting with the tails of tubulins. So to conclude, tubulin tails are important for the binding of different maps to microtubules. Yeah, the different maps exhibit quite a diverse binding behavior to the microtubules. So every single map that I studied, yeah, interacts differently. And yeah, solid state and solution state NMR are tools that give deeper insight into these dynamic interactions. But of course, there's quite a lot to study, and there are quite some experiments that we still want to do for this project. Yeah, and with this, I would like to acknowledge some people. So first of all, my supervisor, Mark Baldus, my master student, Hanneke Smedes, and a postdoc in our lab, Salima, who is helping me a bit with the solid state, and of course my whole group, but also Hugo van Ing, who helped me with the solution state 3Ds, the whole Bloembergengebouw, so that's the building where I'm working, Anna Agmanova. With her, I'm doing some turf experiments on the binding that I didn't show here. And also, I want to thank Caroline Morris and Mamata Banguera, who actually got the same QuiUM structure of MAP7 on microtubules, but just a couple of months too late to be the first. Yeah, and with this, I want to thank you for your attention.